and welcome to the 2020 Brain and Transcranial Photobiomodulation Virtual Summit. I'm your host, Dr. Joe Doduro. My guest is Dr. Paulo Cassano. He's an assistant professor of psychiatry at Harvard Medical School and director of photobiomodulation at the Massachusetts General Hospital, Depression and Clinical Research Program, and principal investigator at the MGH Center for Anxiety and Traumatic Stress Disorders. He's received both his MD and PhD in clinical and neuropsychopharmacology from the University of Pisa in Italy. And he pursued postdoctorate studies in mood and anxiety disorders at Harvard. And he's had multiple studies on transcranial photobiomodulation with near-infrared light for anxiety and mood disorders, and including some translational studies aimed at discovering the mechanisms of actions. His groundbreaking projects have demonstrated this wonderful efficacy in treatment of psychiatric disorders, and he's gained considerable attention, being featured on CNN, the Washington Post, Boston Globe, across the nation. He has co-authored more than 50 publications, and he's the associate editor of Photobiomodulation, Photomedicine, and Laser Surgery. My extreme pleasure to welcome you to the summit, Paulo. Thank you for your time.
Thank you, Joe. Thank you for having me. It's a pleasure.
One of the things is that you've got 2009. When did you start to get in, to come from a medical doctor to PhD, to study lasers or photobiomodulation? How did it, what was the impetus, if you will? How did you get started?
It was an accident, really. So by the time I finished my residency training at MGH McLean Hospital in 2009, I was very interested in international psychiatry and community psychiatry. So I'm still to this date a community psychiatrist, and I still practice, see a large population of Latinos for mental health in a community clinic. And at the time, as I was starting this, I realized that my patient didn't like medication. So they didn't like what I had to offer. So I wasn't going too far. So at that point, I decided that something else should be done. And at the same time, I was continuing my research. My background is in depression and anxiety disorders at Mass General Hospital. And there, our group was finding this interesting new discoveries about brain metabolism and depression. And also the new studies on stroke and photobiomodulation conducted by Phototerra were largely publicized in The Economist, and so we found out, wow, this sounds really interesting, and we find that there's a low brain metabolism in depression. So they find me within the program and say, hey, Paulo, would you like to study this? And he said, well, I didn't know anything about it, about photobiomodulation, but I thought, well, maybe that's what I'm looking for, and that's what my patients would be interested in doing. So that's how it started.
I like it. Now, what we found is that the majority of the researchers that we talk to, when they first find out about photobiomodulation and they reported effects, they're quite skeptical. They have a lot of doubt, like, well, this can't really be true. Well, what did you interpret it as? You were more positive then, huh? You said this might help.
Well, my thinking was this might be acceptable to the patients, because this has been a discovery then moving away from or moving on also from the community to the wider population of patients with mental illness, even non-serious mental illness, that there is generally some hesitation to use medications. So as a psychiatrist, when people come to see you, you know that they are ready but that is 25% of the people that suffer of that disorder. And so the fact that we were exploring new treatments outside that conventional path, I think that was interesting and worth pursuing.
Lovely, lovely. Yeah. So I mean, so what you're saying is that a small percentage of the population is going to go to a psychiatrist, right? And then that population, there's a percentage of those people that won't do the therapy that you tell them to do. So basically, to have an impact on the disease state that you're studied, you had to come up with an alternative model. So I follow you.
Absolutely. And by the way, Joe, it's not just that a small percentage comes to the psychiatrist. It's also that even when they go to their primary care and they are offered an antidepressant or psychopharmacological intervention for a mood and anxiety disorder, only a small portion of those patients actually accept it.
Oh, this is I'm excited to let it fall from your lips. Let it begin. Let's look at your slides.
Sure.
Wait 1 second. What do we hope to learn at the end here? What will somebody that watches, what would be the takeaway message that you think, Paul?
The takeaway is that photobiomodulation might be more than one thing, I think.
Okay.
But let's see. All right. So the title of our presentation is Brain plus tPBM. What dose should we use, and how do we reconcile findings? And this is really the lessons from the depression trial. So these are my disclosures. Of note, I'm the co-founder of a company. However, none of the information that is here presented refers to the devices of this company, and I'm not employed by this company. Well, so let's say if our presentation was about transcranial electrical stimulation, if I told you that, would you know what I'm about to talk about? And is it electroconvulsive therapy, for instance? Is it transcranial direct current stimulation, or TDCS? Is it transcranial alternating current stimulation, TACS? Or rather a cranial electrotherapy stimulation? So you can see that within an umbrella, there are different modalities of treatment. Now, we are talking about one entity, which is transcranial photobiomodulation. So let's go and explore that. So I'll spare you our first study, Elite 1. I can only tell you that we came out of it after the bankruptcy of Phototera, which was the company that was actually sponsoring the study. So back then, we were left with a study that was truncated, deviceless, without support of a company. Fortunately, Mike Hamblin suggested that we use the device that you see in this picture, which is the Omnilux NUU device. We were a little skeptical, to be fair, and this was actually an LED wrinkle cosmetic type of device, and we were coming from a true laser device in our previous study. But we had no option, so we went with that, and we started our study with 21 subjects for depression. This was a although small, well-designed, double-blind, randomized, parallel study with 2 sessions per week, 8 weeks follow-up with the 830 nanometer. And this is the device here. We did the dosimetry. We found that, as expected by the randomization, the patients were equally distributed in sham and active. You see here in the circle or squares in red. And there were a few patients that either were lost to follow-up or did not complete. So at the end, this was equally distributed in the two groups. So those patients were not included in the completer analysis, but in the last observation carry forward they were. When we look at the baseline, the groups were fairly comparable. Generally speaking, these were middle-aged patients, and the depression severity was 20 if you look at the MD-17 here. However, despite the moderate severity of the depression, there was a difference, statistical significant difference in the number of episodes, previous episodes of depression, which seemed to be higher in the people that were treated with the actual treatment. About 30% of subjects in both groups were also on antidepressants, and so this for them was an add-on of transcranial photobiomodulation. When you look at the scores, here in red is the active treatment over time. On the Y is the depression severity. These are the weeks. You see that there was a significant decrease of depression severity over time compared to the blue that was sham. 3 analyses were conducted in this study, and 2 out of the 3 showed significant results. This is one of those, which is the baseline carry forward analysis. And also, even more obvious, the finding for the completers, where the effect was also significant and reached an average on the Hamilton D for depression scale of 7, which is considered a remission score. So this, by all means, were very promising, although preliminary results from our related 2 study, even with this wrinkle treatment device, LED device over the counter. When you look at the side effects, here you see the column of the active and the sham. We looked at different side effects here listed on the left, and we look at the prevalence based on the number of patients who experience. You can clearly see that headaches and also some strange side effects like strange taste in the mouth or ringing ears were more common in the active than the sham. And overall, the prevalence of side effects was more common or higher, I should say, in the active as compared to sham. This was another another element that was supporting a potential effect, neuromodular effect of the light in this population.
Paolo, one question about this, because I'm asking, some of these things that they came up with, you suggested these? This was a list of possible side effects that they clicked off, or is this things that they came up with? Because I want it to be like one of the drug ads, you know, you can have, you know, all these different symptoms. I mean, did you make these people aware of possible side effects, or are these the things that they brought up?
So in this study, we're using 2 methods to track side effects. So one was, clinician report. So clinicians would typically ask during an assessment, did you have any problems? And they would report any new onset event and or any worsening of pre-existing events. However, we also had a structured scale called SAFT that was typically used for side effects from antidepressants. And we used that scale, and that's what we are reporting here. so it's a broad spectrum of manifestation that you could have. And on this scale, we then went back and looked whenever there was a change of 2 points in the scores, and we consider that as a positive, as either a new onset or a significant worsening of a pre-existing condition. And that's what you see reported here.
Thank you. Thank you. Because most of them are always saying, well, we have no side effects. We have no side effects. And then we have Harvard saying, well, we got a lot of effects. They may not necessarily be side effects, but they are effects. Go ahead.
Yeah, so the way I would reconcile that is that, if you look at all this strange or unusual effects, they're really short-lasting. The headache or the strange taste in the mouth or seeing kind of vivid colors is really for some few people right after the first treatment, and they last maybe 30 minutes or so. So, it's I can see that, and it happens also in my clinical practice, unless you really ask specifically and you pay attention, people are not going to report it. And this is not anything really troublesome.
Got you. Thank you for that.
Yeah. So we were quite excited. A lot had happened on the ELITE 2. It wasn't just the efficacy. It wasn't just this side effects which we really didn't expect, which intrigued us and made us think that something was really produced, some true neuromodulatory effect, but also odd benefits like increase in sexual functioning, stories that we're hearing from our patients, I remember one of our patients that was over 60 was kicked out by his girlfriend because it was too demanding, and so clearly a lot had happened. So we were excited, especially now that we were going to a confirmatory study, the ELITE 3. We had 2 sites. We had finally a true transcranial device. This was still an LED device, but made to shed light, as you see here in the picture, on 4 different sites, not only F3, F4, where the dorsolateral prefrontal cortex is, but also the frontal pulse. We had a larger sample, still double-blind, randomized, also better design to really create more power, if you wish, and I'll explain that in a few slides. And we still had 2 sessions per week, still 8:30, and we were using the parameters that we had confidence in. So here is the design. We were randomizing subjects to treatment here in red, and actually sham here in red, and to treatment in green. And then the sham non-responders were re-randomized again for treatment or sham. And so that had an important advantage, that at the end you can actually cluster these groups and reuse the patients. So these patients that were sham non-responders were reused, and so you can use the sham phase here in the second phase and the sham phase in the first phase, and same thing for the treatment phases, therefore increasing the overall sample, statistical sample. So this was a design from Maurizio Fava, who's actually the chair now of psychiatry at Mass General. So we randomized 49 at Mass General and with Dr. Iosifescu at NYU, New York. And here I'm going to present some preliminary data from this sample. Here you see according to that structure that I show you, some subjects receive near-infrared light in phase 1 and phase 2, some receive sham in phase 1 and NIR in phase 2, and some sham and sham. Now, this was a smooth sail, I should say, or a smooth ride, this study. Yes, we had the occasional patient that was, in the phase 2 showing a great response, and we were hopeful, and we said, well, maybe this is one of those that went from sham to NIR, but we didn't have those extraordinary side effects or sexual functioning changes. At the end of the day, when we completed the study, and these are again the HEMD scores plotted over time by visit, this was flat. So this was a big letdown. We had presumably used the same dosimetry, and yet if you look here in blue, the Hamilton Depression Scale was NIR and IR, both phases, was worse than sham and sham in green, or sham and NIR. And when we look at the other scale, same thing. Basically, the lines were mingling and no difference. So we went back and we scratched our head and thought, well, what happened here? This was related to on one column, these were the parameters used. This was related 3. Everything was very much the same, maybe a little less total energy, 2.3 kilojoules in elated 3 per session as opposed to 3.4. And then the duration was in elated 3 was 6 weeks instead of 8 weeks. But it didn't really make a lot of sense. So one thing that we had noticed, though, was that these 2 sites, which were supposed to be on a 3 F4 of the device, these LEDs were actually on here oftentimes. It was a rigid, a little clunky device. And we thought, well, then if that's the case, we probably just deliver half the amount of energy. Ooh. And that made a lot of sense to us. And that was really the first conclusion here. 2 very similar samples of depressed patients enrolled with the same inclusion-exclusion criteria. You give them half the dose, well, they don't respond anymore. So dose matters. Now, one thing I want to tell you is also, Joe, that during that time, Luis de Taboada from Lycure and Neurothera and I were really thinking, well, yes, we're doing CW study because we had done it before in Melody 2, and that really makes more sense. But what about PW? What about pulse wave? And we were so intrigued and thought that this would be even better than CW. And we had started a small pilot in healthy subjects, which we call NR flow study, with exactly the same device that you saw there. And we wanted to record if there was any difference quickly, so that if we needed to, we could move to a different phase with PW. And so this had been going on while we were performing the Elite 3 study. So this is the structure of the NIR flow study in healthy subjects. Basically in yellow here you see 3 sessions in 3 separate weeks. First continuous wave, then sham and pulse. The subject didn't know what they were getting. So it was just single blind. And most importantly, we were doing EEG before and after, and a diffusion correlation spectroscopy. So we're interested in looking if there was any electrical changes in brain waves, brain activity, and any blood flow changes before and after.
This is going to be exciting. This is going to be exciting. I'm aware there's a big drum roll here, at least on this side. Go ahead.
Okay. Well, so So we were wondering here, why even bother? This is not leading anywhere, and we've seen that the dose was too little. This was still our sample, fairly young subjects. Here you see the age on this column. And then I get this email from my colleague, Diane Sitnikova, Tatiana Sitnikova. And she tells me, Paolo, this is great. This is working. And in fact, she shows me this graph here, looking at the effect on power spectrum density. And here is before the light in blue, and this is after the light in red. And she shows me that at 40 hertz for gamma activity, there's a strong effect of this transcranial light. This was exactly CW. So this was like, what? So now we had the same dose in depressed patients, didn't produce any effect with the same device. And in healthy subject, it did produce a significant change and did actually have a boosting of gamma brain waves, gamma activity.
Let me understand this just so that we're clear. You gave them constant wave activity and saw both, it appears to be a 10 hertz and a 40 hertz increase in their power.
Well, 10 hertz, so here you have to think that these are eye closed resting state EEG. So you're going to see this peak on eye close. And so if you close your eyes, your alpha waves will increase. However, you can see here that there's no separation in between before and after, the blue and the red. So this is very much a physiological change. Where it's unexpected is this change here, which is more pronounced over the 40 hertz.
The thing that's amazing to me is that you didn't put any pulsing in, yet it changed the brain at a certain frequency. That's very interesting.
Absolutely. Absolutely. Even that, I'm not showing here, but the tracing for open eyes showed also beta as a significant change. So this is quite surprising overall, to say the least. We continue to explore this, and we went to look at the tracing of cerebral blood flow. After all, that's what we were looking primarily at. So here were the tracing for the short separation, which is more the blood flow on the superficial tissues with the diffusion correlation spectroscopy, which is similar to FNIRS. And you see pretty much a flat signal. Here in yellow is the treatment phase, which is what you expect. But CW was pretty flat too. So now we had, and this was superficial, this is deep into the tissue. Again, deep separation, still flat sham. A small increase with, which you would expect with the actual CW. However, if you look at the logarithmic scale, this is a minimal effect and was not significant at the end of the day. So now we had the same dose in healthy subjects. Now it had an effect on EEG, but had no effect on blood flow. So that was, again, mind-boggling and certainly new for us. Well, you have to think and go back to the primary question. Yes, we were interested in blood flow, we were interested in EEG, but we were doing a depression study. And we wanted to know if we had to use the CW or the PW. What should we go for? Do we need to change any point in the study? So we told you about the CW. It turned out that PW was just a flat result. So at that point I said, wait a minute. I called Luis de Taboada at Lightcure and told him, look, I mean, we need to do some dosimetry here. We really don't know what's happening here. This is just the opposite of what we were expecting. And so we did. And it turned out that PW here was had been set by basically leveling the maximum intensity. So because this was a 33% duty cycle, PW was 1/3 of the dose of CW. So that made sense at that point. Now we were facing a dose effect in healthy subjects. If you were lowering the dose by 1/3 in the same sample of healthy subjects, you were actually losing the effect on brain activity.
So just for me, because I'm not the math guy, but a constant, continuous wave or constant wave LED device is going to have 100% on, right?
Mm-hmm.
And then if you do a pulsed wave, one of the things is that we would think about is that it's going to turn on and off at a certain frequency or time. But what you're talking about is a duty cycle, which is going up and down versus on and off. Am I correct in that?
No, the duty cycle is exactly what you mentioned before, the on and off. So if you are 33% on, at the end of the day, and if you're using as your peak intensity the same intensity of CW, then you're just delivering 1/3 of the light because you're only on 33% of the time.
Right. But that would be a frequency of what?
The frequency used here for pulse was 10 hertz, and the time on was 33% of the time.
So I try to put that together because that what I understand it to be would be it would turn on and off 10 times a minute.
So the 10 hertz is referred to the frequency of turning on. You're right about that. Then what you can do is whenever you turn on, you vary the length of time that it's on. And with that length of time on, you can actually vary what we call the duty cycle, which is the total exposure time over the 100% time of treatment.
And that really affects it differently because of the different ways that that's going to come in. How is that? What's the rise of that cycle and the downside of that cycle? And you know what I mean? What is the maximum? Like you're saying, you're getting it 33% is going to be the maximum. That's the dose that you're getting when you turn it on. Is that what I'm understanding? The curve is like that?
Yes. My understanding here is that you have to think about this mostly as basically a square wave. And so you go on and you vary. It's more kind of a switch on and off. And there are two elements. How often do you go on? And for how long every time you go on, you stay on. And that will matter. Here, the problem was, I don't think was how often you go on, the 10 hertz. It was rather how long you stay on, which is then just 1/3 of the time.
I see. That's it. It gets a little more complicated than just a regular chess game. Go ahead.
Yeah, no, it was complicated. I agree with you. So this was, as we were conducting our depression study, the results from our healthy subject study. So to summarize here what we have learned that if you look at similar samples of MDD patients and you decrease by half the dose, you're going to lose the effect. So dose matter in a clinical study. It's really important to get that right. Also, we found that the same dose that in the MDD sample was producing no clinical effect with the same device, this was CW, was actually producing effects on the healthy subjects, so in the healthy subject sample. And so this has implications because we look at healthy subjects, it's easy to do those studies, it's fast, it's easy to recruit, but can you always translate what you're seeing there? And also this could be a reflection of the fact that we were looking at specifically biological and neurophysiological changes rather than clinical changes. And so how much one translates into the other, that's also important.
It's important, very good point.
So we also saw that the same dose in the healthy subjects had an effect on EEG, but had no effect on cerebral blood flow. Now, this could be a limitation in the technology that we use in cerebral blood flow. If there's anything that we know about photobiomodulation is that it improves blood flow, vasodilation, and there are studies from Francisco Gonzalez Lima's group on blood flow, and we are very confident of that. However, at this dose, you had one effect and not the other. And if that's the case, that challenges our unitary view of photobiomodulation. Because if with different doses, or if you need different doses to reach a certain neurophysiological effect, let's say at a lower dose you reach an electrophysiological effect, and at a higher dose you reach an effect that is also an effect on hemodynamic, on brain, then shouldn't we use the same umbrella like for electrical stimulation, where we actually define the treatment based on the physiological effect rather than the principles that we use? So we know that if you deliver a lot of electrical stimulation, you can provoke a convulsion, and that's ECT, which is different from a focal effect with the lower threshold, which you use for TMS, which is different from an even lower effect, which just increases excitability, which is TDCS. So maybe photobiomodulation is not one thing when it comes to its modalities and its neurophysiological effects. Finally, again, one more time, even in the healthy subjects, we see a dose effect, even in the electrophysiological changes. So there's no question that dose matter. I'm not sure now if I answer the question that I started with. It's like, how do we define dose? I can tell you that dose matters, and that's something we know. And if we are going to study the effects of photobiomodulation, we need to do dose finding studies and be aware that at any of those steps, we might lose translation, if you wish, from a neurophysiological finding to clinical finding, from healthy subjects to clinical population. And that doesn't mean that there's no potential, that the treatment is not valid, or that there's no future for photobiomodulation. We'll certainly have negative studies just because it's challenging to translate through this chain. Finally, this is our group, the depression program, and I'm thankful for the contribution of many. And this is also the division of neuropsychiatry. Now we are living virtual, so this is how we are as a group today. Thank you.
Well done. Thank you. Thank you, Paolo. Thank you for that. You know, I still think that the questions that you raised and that you know, your evaluations are still so, you know, so poignant. That if you make an elegant experiment, something will be revealed to you. You follow? So that teased out a lot of that. But, you know, it made me think for a moment about the surgery. And if you do surgery, they try to tell you this is how you do it. You know, you go this much of a cut, and then you open it up, and you go in this far to do that. And in reality, sometimes when we're using transcranial photobiomodulation, it's not the what that we do, but we need to describe the scalpel better. Because sometimes you can't really cut if you got a rubber scalpel, or if it's not thin enough, or if it's not strong enough, or if it's not, you know what I mean? You're not going to be able to, it's not so just much about penetration, but the characteristic of the tool that you're using. And I think you pointed that out kind of, you know, in a good way. What do you think of that perspective?
Yeah, absolutely. You have to understand what are the characteristics of the tools you're using, whether it's using pulsing, what intensity are you using, what wavelength are you using, and what's the total energy, where are you shedding the light to begin with. If it's multiple sites, you're shedding at the same time. And that's the challenge, that there are so many variables that can change, and how do you test all those variables at one time? It's virtually impossible. And I would add to that you need to know the characteristics of the person and of the head, of the brain that you are targeting as well. So that really can create the best match.
You know, there's no doubt that you're one of the first studies that actually looked at the pigmentation of the clients or patients or subjects that you were studying. They gave you some withdrawal to realize there may be some scatter and some stuff non-penetration. So I think that, as I said, it's a quite elegant study, and it did reveal information about what you were doing. So there's nobody that is looking at it like that. Can you speak to us now about what you see in the future? I mean, you're going to be around, let's say I'm going to be around with a healthy brain in 2030 when we do the 2030 Brain and Transcranial Photobiomodulation Virtual Summit, which probably we won't even have to stare at our screens, who knows, but you know, you're involved in this, you're involved in the commercialization of some of these things. Just tell me, what do you think? Because you're the person that we really want to ask. What do you see the future in 10 years? How do you see this interacting between photobiomodulation, the brain, and specifically like your field, which is depression and mood disorders, anxiety?
Yeah, so I think there are two elements here. One is the clinical, and there's no question that in our field of brain disorders, we know that devices are really effective. Electroconvulsive therapy and TMS and vagus nerve stimulation are the most effective treatments that we have, and they're tremendously helpful. Mm-hmm. However, all those have been fairly difficult to access. There are in-office treatments. They're expensive. Some require anesthesia. Some are invasive, like VNS or vagus nerve stimulation. So we need devices that bring that level of efficacy, or at least very close to that, at home. Devices that are portable, devices that are possibly wearable, because clearly patients need more than psychotherapy and medications, and devices can offer a lot. So I think there is a future in creating therapeutic devices that allow us to deliver treatment at home and hopefully self-administration. So that's from the clinical. I think that needs to happen. There needs to be trials that show the feasibility of that. And photobiomodulation is well placed because it's easy, it's inexpensive, and also it doesn't require to wet sponges. It's not complicated. So there is a lot of rationale besides all the science that we haven't looked, the biological rationale as well. That's the clinical. Then there is the health of our brain, which is another important chapter that I think it's going to be the future. In the 80s, we started understanding that we should take care of our heart and mind the health of our heart. Now we know that brain health is crucial. And we are in this different phase, and we're thinking at what to do. And photobiomodulation can be an instrument for all of us in the non-clinical, in the wellness, to keep our brain healthy. And so brain health is the future, because we know that we cannot pay for the needs of an elderly aging population, and we need to keep our brain healthy as much as possible. And finally, there is brain hacking. There is performance, and we've always tried that. Whether it's coffee, chocolate, a glass of wine, a glass of beer, we know how physical activity, how to boost our performance, how to press speed or slow down our brain. And photobiomodulation can be one of those tools. And I think that's part of the future.
So do you predict there's be a time when we can get on our quote unquote smartphone and dial in the mood that we want?
I think personalization is key, and we can have devices that are smart devices. We're gathering tons of data all the time on what we do through our phone, and so it's just right there, ready for us to use and to input into photobiomodulation or some other type of neuromodulation. Whether we will dial in or not, I don't know, and how precise that's going to be, it's hard to tell. My sense is that fortunately photobiomodulation doesn't have an effect like cocaine or anything that would give you instant euphoria, and so that's reassuring, because otherwise what you are kind kind of hypothesizing could become addictive.
Well, but 10 years from now anyway, at the 2030 summit, I believe that we're going to say, do you remember how much we used to stare at our phones? You know what I mean? They're going to, we're already addicted to smart devices. It doesn't matter if it was cocaine, it'd be easier to sell it. You know, if it had that effect, everybody would be buying it. But
sounds good.
Thank you, Paulo, for your time and efforts and all the research that you're doing. Wish you continued success and keep elucidating us as to how this stuff works for better brain health, better mental health for us humans. Thank you.
Thank you, Joe. I'm so thrilled that you put together this platform, and I'm really looking forward to listen to all the different talks of my colleagues and friends.
Thank you.